Hello, in this class we will learn another new technique that is being used in combination with mass spectrometry, which is called fast photochemical oxidation of proteins. This is mostly used in the area of structural biology to understand molecular basis of a biological event, like it can be human disease or it can be any other biological process also. So, the principle of fast photochemical oxidation of protein, which is also defined as FPOP, is the hydroxyl radical based covalent labeling technique that is used to probe protein structure, protein-protein interaction, conformational dynamics and ligand induced structural changes of protein. So, first and foremost thing is we have to generate hydroxyl radicals. It is a free radical. Any free radical is highly reactive and their lifetime is short also. So, in this FPOP, hydroxyl radicals are generated in solution using a nanosecond UV laser pulse, which exists for less than 1 microsecond. Now, the hydroxyl radicals generally whatever is produced, they are very reactive. So, they rapidly and irreversibly oxidize solvent exposed amino acid side chains, ensuring the snapshot labeling without structural rearrangement. So, therefore, this hydroxyl radical based oxidation that reflects the native conformation at that instant, not the equilibrium structure. So, basically kinetics, structural changes along with kinetics, along with time frame, we can monitor using this strategic approach. Now, biological function obviously meaning any molecule, we are discussing about protein. So, biological functions of protein obviously determined by its 3-dimensional structure, which is higher order structure HOG, and that is the structure monitoring we are talking about using this platform. Now, protein footprinting is an assay that provides the structural details associated with its function, such as identifying the site of ligand interaction, solvent accessible surfaces of the proteins, the location of structural modifications, the structural changes associated with the biological event. Now, we have studied HDX, isotope exchange based mass spectrometry, that is also used to understand the molecular conformational changes associated with a biological event, and like HDX, FPOP also monitored through mass spectrometry platform is an analytical technique to explore protein footprinting. And as I said just now, the protein footprinting includes anything, any structural change associated with a biological event. Now, FPOP coupled to mass spectrometry has been employed in solving several important problems, including mapping of epitopes, that is locating the part of antigen that antibody binds to, following protein aggregation, site of small molecule binding, measuring ligand binding affinity, the association dissociation, monitoring protein folding, unfolding event, and determining hidden conformational change invisible by other methods. So, let us talk about the principle of actions, how to generate hydroxyl radical. That is the first and foremost thing required. Hydroxyl radicals can be generated by the decomposition of H2O2 by Fenton chemistry, but that Fenton reaction takes minutes to complete, and during that time protein might be over labeled due to labeling induced changes that happen in protein conformation during the footprinting, that is at the first step. So, another strategy approach is homolytic cleavage of H2O2 using UV radiation, say UV radiation up to 48 nanometer. Now, in that case UV lamp can be used. Now, in that case if we use UV lamp, that requires large concentration of hydrogen peroxide, 15% weight by weight, and that in presence of that large oxidizing agent, because hydrogen peroxide is oxidizing agent itself, so there may be a direct oxidation of protein by the reagent, besides the footprinting that we are talking about caused by OH dot. So another strategy is to use KRF excimer laser that produces OH dot in nanosecond scale and works well with the low concentration of proteins, 15 millimolar, 0.04%. And prior to laser irradiation, the protein is mixed with hydrogen peroxide H2O2 and a radical scavenger that must be there along in the system itself, which is basically an amino acid. Now, OH dot, once it's generated, undergoes Haber-Wey's reaction in competition with the self-quenching to reform H2O2. That means, once we produce H2O2 by the dissociation to 2H dot, then OH dot can attack another H2O2 molecule, generating H2O and HOO dot. Then HOO dot can attack H2O2 molecule again, and then that may produce one molecule of water, one molecule of oxygen, and one molecule of OH dot. Now, these two OH dot can combine to give back hydrogen peroxide again. Now, this OH induced protein oxidation by abstracting a hydrogen atom from protein, which results in the formation of protein radical, that further undergoes a series of subsequent reactions. As we know that once a radical is formed, then it goes literally chain reactions to generate products with a stable oxidation. Now, rate constant for the reaction with which amino acid reacts it's in the range of 10 to the 7 to 10 to the 10 molar inverse second inverse, so high. Now, OH dot reacts with generally 14 out of 20 amino acids, and majority of the modifications leads to addition of 16 Dalton. In absence of a scavenger, the radical lifetime in FPOP is limited by the recombination reactions, like the recombination 2OH dot forming back to produce H2O2, and can extend up to 100 microseconds. Now, allowing a protein to react for this time, leaving the protein in the radical environment for 100 microseconds may lead to unfolding, exposing buried residues to solvent molecules. So, adding a scavenger is very much important, which will kill the radicals, and so that we don't want unwanted reaction happening, right. So, addition of a scavenger, 20 millimolar glutamine, to the protein solution effectively shortens the lifetime of primary hydroxyl radical from 100 microsecond to 1 microsecond. So, limiting the modification to reactive and solvent exposed residue in the native conformation. Meaning, we are in a mode, we are planning in such a way that we are hoping in such a way that OH dot should react, but we don't want overreaction, right? Over labeling, we want to avoid. So, for 15 millimolar H2O2, we found the initial concentration of OH dot is in the range of 1 millimolar, 0.95 millimolar. Now, FPOP modified proteins usually can be analyzed by bottom-up proteomics strategy, bottom-up proteomics approach to obtain subregional residue-specific structural information, that is footprinting. And this is the place where it differs from HDX, because in HDX labeling is done by the replacement of hydrogen by deuterium, and that labeling is not very stable. If there is a chance, meaning if the environment is filled with normal water, then that exchanged deuterium can go back to the solvent. So, it's a reversible process, but in case of FPOP, whatever reaction has happened, that is irreversible. So, now the modification cannot be changed, and to find out the site of modification and then literally to extract out information related to structure, we can make the use of make use of the platform proteomics, bottom-up proteomics approach by digesting this modified protein through the proteolytic cleavage. Now, optimized exposure of radical is necessary to avoid any radical-induced unfolding of protein, which might lead to the exposure of inaccessible region of protein molecule. Okay. So, now let us take one example like application of FPOP in amyloid protein aggregation. So, we know that the aggregation of amyloid beta protein A beta is one of the primary pathogenic events leading to Alzheimer's disease. Now, characterizing the products of amyloid beta aggregation is challenging because of vast heterogeneity and transient nature of the intermediate aggregate, because it does not have a very stable structure, it is very much flexible. So, characterization of any aggregate is not so easy. So, FPOP was used to monitor the amyloid formation and solvent accessibility of A beta 42, the 42 amino acids peptide of A beta at the global peptide level, at the amino acid residue level, and so on and so forth. So, FPOP footprinting captured solvent accessibility of amyloid beta A beta 1 to 42 intermediate. Irreversible modification by FPOP should not be affected by A beta amyloid beta aggregation. This is important. Whatever modification is happening with FPOP is irreversible, but that irreversible modification should not affect the amyloid beta aggregation, that the biological process which we are actually studying. So, this is a bit tricky in case of FPOP, which wasn't there in case of HDXMS. Now, A beta 42 aggregates in various oligomeric states monitored by FPOP showed extensive modification of amyloid beta 1 to 42 residue monomer owing to its intrinsically disordered structure, that is in this picture, figure A, where we have several of the amino acids detected from the same molecule, and this is basically owing to intrinsically disordered structure. So, several different conformations are possible that can ionize with different charge states, which is visible under mass spectrometry condition. Now, amyloid beta 42 assembles and reorganizes into higher order oligomers, and modification of A beta 42 decreases owing to the formation of secondary and tertiary structure, as we see in the figure B and C. So, meaning moment aggregation is happening, it is an ongoing process, then the structural flexibility of the amyloid beta polypeptide chain decreases, and that is reflected in the probable charge states, disappearance of probable charge states or multiple charge states as we go along with the time. So, that is being seen in figure B and C. Now, in the end of aggregation, like after 48 hours, as we have shown in the figure D, the modification extent decreases modest level, indicating the formation of compact amyloid fibril with a core structure that resists the modification. Now, fit of a plot of modification as a function of incubation time shows sequential formation of the major species A beta 42 during aggregation, as in figure E. So, it basically shows a plot of modifications as a function of incubation time. So, different times we have monitored, they have monitored, it is not our work, it is actually taken from this paper that came in ChemRes in 2018. So, it basically shows that the different type of modifications that are there across different time interval during the aggregation process of A beta amyloid. Now, interconversion of the representative A beta species by stimulating their concentration changes as a function of incubation time are shown in figure 5, in figure F. So, like if you see that the figure F, the concentration in monomeric equivalent change of representative A beta 42 species, M stands for monomer, D stands for paranuclei, D star stands for protofibril, and D double star stands for fibril. And this is these are the different type of oligomers that are being monitored in terms of its formation across different time interval in the aggregation step. Now, modification of N-terminal peptide 1 to 15 residue that remains high with a little variation as A beta 42 aggregates. So, literally there is no change. If we look at the figure G, in the N-terminal part, there is no change basically of it in terms of percentage of modification across time, which is why it is being stated here that the modification of N-terminal peptide 1 to 15 remains high with a little variation in A beta 42 aggregates. This indicates that N-terminus basically remains structurally flexible and solvent accessible. So, its structural geometry conformation hasn't changed much across the oligomerization process. With that means it's actually participating in the aggregation process to a very little extent. So, N-terminus A beta 42 is disordered in a variety of oligomers and in mature fibril. So, its disorderness stays back. Now, critical role for hydrophobic C-terminus in promoting self-association are highlighted here in the figure H by a dramatic decrease in FPOP. If you follow this green line in figure H, there is dramatic decrease in FPOP across time, FPOP modification of monomer to fibril. So, H6 in figure I and the ratio F19/20, how these two at the residue level, how the structure is changing, that has been monitored through FPOP. Okay. So, H6 shows a constant FPOP modification during aggregation, indicating little change in its solvent accessibility and conformation. So, it is basically a straight line in case of I, figure I. Whereas, F19/F20 residues responsible for modification observed for central domain as in figure J, right. So, finally, it was proposed that the interactions of F19/F20 contribute a driving force for A beta 42 aggregation by serving as a hydrophobic nucleation interface. So, in a nutshell, the entire A beta aggregation process can be very well studied using FPOP approach, FPOP coupled to mass spectrometry platform. Now, I will take you to another application of FPOP, that is in vivo application of wedge dot labeling. I showed in vivo application of hydrogen deuterium exchange in case of RBC. Here, this is another example where in vivo application of wedge dot labeling. So, permeability of H2O2 across cell membrane via diffusion or through channel proteins made in the in vivo application, of FPOP feasible. Until unless H2O2 goes inside cell, we cannot generate wedge dot there itself and make use of this reactivity of wedge dot to react with different conformation of proteins that are present in the cell. So, the main motto is we have to generate wedge dot inside the cell itself, and that is possible because hydrogen peroxide penetrates through cell membrane and then once it goes in, then we will expose the cell containing hydrogen peroxide with a laser, which will generate wedge dot the way I explained to you, and then that wedge dot will participate in different reactions. So, in order to study protein conformation in a living cell, FPOP footprinting method was used in cell, and oxidative modification of protein in various subcellular compartment was studied in African green monkey kidney cell, which is called Vero cell. Now, after optimum incubation of Vero cell in H2O2, live Vero cell we are talking about in H2O2, followed by pulsed laser exposure, the cells were lysed and proteome pool was subjected to proteolytic digestion using trypsin as a proteolytic enzyme. Now, next is proteomics approach, bottom-up proteomics approach. So, nano LC-SIMS or LCMS analysis was performed to identify and characterize site-specific modification and quantification of oxidized protein in the cell. So, to probe the solvent accessibility of protein within a crowded environment inside cell, oxidative modification of actin was investigated. Now, in the in vivo study which appeared in this paper, Analytical Chemistry 2015, volume 87, page number 7971. This is the starting page. Now, as I said, the obtained protein profile of oxidation pattern of different residues of actin was compared with previously reported in vitro result. That is always the basis. We have seen similar event in case of hydrogen deuterium, in vivo hydrogen deuterium exchange based mass spectrometry. There, in fact, we have compared the literature information that we got over the years from different scientists across the globe in the structural biology field, and then compare that in vitro result with the one in vivo that we had. Here also same approach. The study of actin conformation in cell, the solvent accessible surface area, which is called SASA, of both open and closed conformation of homologous bovine actin were compared with the obtained cell in cell FPOP data. So, the observation was, although actin is likely to be present in multiple conformation inside a cell, but the in-cell FPOP data and its comparison with the SASA of actin suggested that most abundant conformation of actin in cell is open structure rather than tightly packed one. So, this in vivo approach, whatever I have explained, I have taken the gist of this paper which appeared in Analytical Chemistry 2015. Okay. So, with this I will stop today's class on fast photochemical oxidation of protein based footprinting of protein molecules to understand different biological events starting from its structure, structural transition and so on and so forth. In the next class, which would be going to be the last class in this course, we will discuss about something else which is having direct application in the industry. So, with this note, I will end the class, today's class. Thank you.